All right, good afternoon, everybody. Before we go on, I wanted to note there's a room change for this author talk on Monday. It's been changed to Davis, which is a bigger place. I guess they're anticipating a bigger crowd. But if you have some time, you might want to check it out. It should be interesting, the author of this book. Today I want to go inside the virion and start looking at how they're put together. And it's always a hard decision what to do first, the outside, the capsid of the virion or the nucleic acid. And I choose the nucleic acid because that's what runs everything. So it's a good thing to have a good understanding of the boss before you get going. So today we're going to talk about genomes, what they're like, how they're constructed, and how you can manipulate them. Now, last time we talked about the breakthrough that was made in the 1950s, that the viral nucleic acid genome was shown to carry the information that's needed to make new viruses. And as I said the other day, that this is something that seems obvious now, but that discovery was one of the building blocks of molecular biology. It was made using two different viruses, a virus of bacteria called T4, a bacteriophage, and that was the Hershey-Chase experiment, which we showed in I think the first lecture, and which we'll look at again later today, and also experiments with tobacco mosaic virus, the first virus to be discovered. And the experiments that were done were to take the RNA out of this virus particle and insert it into cells, into plant cells, and show that that's enough to start an infection, proving that the nucleic acid is where all the information to make new virions lie. And again, it's not trivial because at the time people thought the protein might be the genetic information. So these are important experiments. So perhaps the biggest surprise, although there are thousands of different virions and apparently infinite complexity of infections of different hosts, there are really only 7 genome types, as we said last time. There's a finite number of genomes, and in fact there are 7. And that's great. And they're either DNA or RNA. And we're going to talk about that today. The composition and structures of these genomes are more varied than any in any other kingdom, bacteria, animals, plants. They're just amazing. The structures are very diverse, and so are the coding tactics, and we're going to start touching on that today. And nearly every possible tactic for encoding and decoding, decoding meaning translation into protein information, nucleic acid, can be found in these viral genomes. Very diverse. And for molecular biologists, they provide an endless source of work to do. Viral RNA or DNA genomes are structurally quite diverse, as you'll see in a few moments. They can be linear molecules, they can be circles, they can be segmented, that is in pieces, with each piece having one or more gene on it. They can have gaps in them. They can be single-stranded of plus polarity or minus polarity. And they can be ambisense, which means they have a little bit of plus and a little bit of minus, as you'll see a bit later. They can also be double-stranded. Some of the genomes have proteins attached to them, as if all these configurations weren't enough. And some have cross-linked ends of double-stranded DNA. So an amazing variety which we see in any other kind of animal, plant, or bacteria. There's even some DNAs with covalently attached RNA. So what does this all mean? That's the question I see very often. Why are there so many different genome types? And is this something that will give us some clues about the biology of the virus? In other words, the genome type, the particular configuration, how strange it is, or viral evolution? Is one genome type more advantageous than another? And finally, the most interesting or important question, do we have to memorize all this stuff? Now, these questions, most of these are difficult to answer with the exception of the last one. That's an easy question to answer. So I'm going to just go through some of these issues, but I'm not going to have any answers for you. And maybe at the end of the course you'll have some ideas. Why is a given structure used? Why is it good for that particular virus? The structure of the genome is a reflection of how the virus replicates and how it packages the nucleic acid, and you'll see some examples of that. So particular structures at the ends of RNAs or DNAs are important for packaging, so that's why they evolved, but they're not present in all viruses. So we don't really know what the selective advantage is in most cases. Why do we have both RNA and DNA in viral genomes? You know, we have a DNA-based genome. Bacteria do, plants do. Why do viruses have one or the other? Well, there's a theory that many, many years ago there was an RNA world where all organisms had RNA genomes. Millions and millions of years ago, of course. And that's called the RNA world. And we think that because the RNA genomes, the only RNA genomes left on the planet are those of viruses, that maybe viruses were around in the RNA world back then. So they're relics of an ancient world. This is purely hypothetical. There's a lot of evidence for an RNA world, but we don't know if there were viruses there or not. And if this is true, if there was an RNA world and the viruses were there, why did they switch to DNA? Or why did some of them switch to having DNA? There must have been some evolutionary advantage for that to occur. So these are all interesting questions that I can't answer for you. And in the course of learning virology, maybe you'll get some information which gives you ideas about this as well. But I can tell you should memorize those 7 genome types. That is one thing, if anything, that you should do, because it will make the rest of the course totally easier if you know what the 7 genome types are and more or less how they go to RNA, as we'll see in a moment. So what type of information is in a genome? Well, gene products, proteins, and regulatory signals, for the most part, that are needed for replication, for replication of the viral genome, for packaging the genome in a new particle, and also regulating the timing of the cycle. You'll see some viruses do some regulation, which is quite interesting, so the gene products are involved in that. And to modulate host defenses. When we talk about how viruses cause disease, you'll see there's a huge offense-defense movement. A virus does something, the host counters, and then the virus counters back, so there are a number of gene products involved there. And then there are gene products that the viruses need to spread to other cells and hosts. So that's what all the gene products in the genome do. What's not encoded in the genome is perhaps just as interesting to look at. There are no genes encoding the complete protein synthesis machinery. So you have probably learned about protein synthesis in other courses. You know there are ribosomes, tRNAs, tRNA synthetases, termination factors, initiation factors. All of these proteins are supplied by the host. No virus encodes all of them, although there are some viruses that encode a few of them. A few RNAs and proteins involved in translation, particularly these giant viruses like the viruses of amoeba that we mentioned. They seem to have a lot of genes encoding parts of the translation machinery. No virus encodes genes for proteins that are involved in energy production, ATP synthesis, for example, or membrane biosynthesis. Viruses don't do that. They get all that from the host cell. And there are no centromeres or telomeres, which are structures typically found in standard host chromosomes. For the most part, we don't see this in viral genomes. But I think we just haven't found them yet, to be honest. I think as we discover new viruses, we're going to start finding some of this stuff. You know, when I used to lecture 10 years ago, I used to say no virus encodes any part of the translation machinery. And now I've changed it. Some viruses encode some parts of it. So maybe in 5 years we'll have proteins involved in these other processes. So the absence of something in virology doesn't mean that it doesn't exist. We just may not have found it yet. Now, to make your life easier, remember, viral genomes must make RNA that can be read by host ribosomes. So I want to make your life easier so you love virology. When you end this course, you will love the subject for the rest of your lives and not look at it as a chore. So here is one key fact, which I've told you before. I'm a big believer in repetition, and it's a good way to learn. So I'll repeat things many times. If you remember that you have to make RNA, and if you remember those 7 genome types, it will really help you understand the information flow for many, many different viruses. And now here's the Baltimore system, which will help us focus on these pathways. So here the 7 different genome types are shown in the diagram, which shows how they lead to mRNA. And if you can remember this scheme, you'll really do very well. So you might want to just commit it to memory such that you start with it. Here's a good way to do it. You put mRNA in the middle in a box, and then remember all the 7 genome types and think of how they lead to mRNA. And they're not very complicated, and most of them make sense, especially after hearing what I have to say today, you'll see that you can deduce these very easily. In addition to that, of course, you need to know some conventions, which we introduced the other day. mRNA is always the plus strand by convention, and that doesn't arise from any fact. I mean, you could have said mRNA is minus. There's no reason why it has to be plus, but it's convention. DNA of the equivalent polarity is plus strand. And the complements are negative strands. So it's pretty straightforward. mRNA is ribosome ready. It can be translated into protein. That's the definition of mRNA. But not all plus RNA is mRNA. So not all plus RNA is going to be translated. So don't get fooled if you see a plus RNA in a viral genome or in a virus-infected cell that it's necessarily mRNA. There are certain characteristics of a message of a plus RNA that will make it an mRNA. We'll go over those a bit later. So here's a secret to success in virology. There are only a few ways to copy these 7 genomes and to produce mRNA. If you start with a double-stranded DNA, if you listen to what I tell you today, you know there's only one way to make messenger RNA from that. If you start from a single-stranded DNA, you will know there's only one way to make messenger RNA. So that makes it much easier for you. There aren't infinite things to remember. Each way has a unique polymerase, a nucleic acid polymerase, a primer, a template, and termination requirements. And these are things we will look at in 2 separate sessions. We'll look at RNA and DNA separately in a couple of weeks. And each virus family has its own characteristic genome strategy. So what will be good for you to do is to not only remember that one of the 7 genome types is double-stranded DNA, but what viruses, the main viruses that we talk about are in that family. Because that will key you into the way that virus replicates. So here are the 7 classes of genomes displayed in a different way, just so that it becomes more clear. So we have 7 classes, 3, 4, 5, 6, 7 of genomes. We have RNA viruses. We have reverse transcribing viruses that go from RNA to DNA. And we have DNA viruses. So 3 broad classifications, if you will, in which there are the 7 genome classes. So this is a good picture to keep in your head. And down below here are some examples of some of these viruses that we'll talk about. So memorize the 7 genome types and the key virus families that use them. And don't wait until like the night before. It's like every time you come here while you're waiting, you should just think about the 7 genome classes. I'll remind you almost every lecture. But it's not something you would want to do the night before. And if you know the genome structure, you will be able to deduce how the mRNA is made from the genome, how the genome is copied to make more genomes, and the textbook will really help simplify all of this because it has a lot of visuals that make it easy. In Chapter 3 is really a summary of everything that we're talking about today, and all the figures I'm using are from Chapter 3. It's a very short chapter. In that chapter, the 7 genome classes are discussed separately, and then there's a box which has very essential information, which I'll show you in a moment. It's got genome configuration, the degree of dependence on the host for replication, and the gene expression strategies. And these are going to be the main things that we will cover over and over. So for example, this is one of these figures from Chapter 3. So this is for single-stranded DNA genomes. Gives you a couple of examples. It shows you the overall pathway, a little more detail than in the Baltimore scheme. The Baltimore scheme just shows you basically the nucleic acid pathway to mRNA, but we've added production of protein and production of virions here. And then below that are different examples of genomes, in this case with single-stranded DNAs. We're going to go through this in some detail today, but it's a good summary of all of this that will help you understand how the nucleic acid flow occurs, and you'll remember it just by looking at this. So here are the 7 classes. We're going to go through this today again. Double-stranded DNA, gapped double-stranded DNA, so it's a variant of double-stranded DNA, and single-stranded DNA, so 3 types of DNA genomes. And 4 RNA genomes, double-stranded RNA, single-stranded plus or minus polarity, and then single-stranded plus RNA with a DNA intermediate. So we separate that those viruses from these, because they do something unusual. They make DNA in their life cycles, and these viruses don't. Now, when you're done with this course, I think you'll be able to know the answers to these questions. So this is what I really want to emphasize. What are the steps to mRNA production from each genome? So if I say a double-stranded DNA virus has to do what to make mRNA, you'll be able to tell me. What general gene products must function at each step? So to make mRNA from a double-stranded DNA, what enzyme is needed? When in the life cycle is mRNA made? Is it made right away as soon as the virus gets in the cell, or is it made later? Do other things have to happen before mRNA can be made? How is the genome copied? What's the mechanism of copying? What are the proteins that copy the genome? And what are the proteins that make mRNA? Are they the same or are they different? And how is the encoded information in the genome converted to proteins? So this you're going to learn in the next half a dozen lectures or so. And by the end of that, you will know the answers to all of these. And we'll know that before we move on to pathogenesis. So also another useful resource in the textbook is an appendix. There's an appendix in each volume. In Appendix A, Volume 1 is Basic Molecular Biology. And at the end of that volume, there's Appendix A. And it has viral life cycles, facts about the genomes. And I think it's useful to look at these just casually to start relating the 7 genome strategies with the overall life cycle. So today I'm going to give you an abstracted view of how these genomes go from nucleic acid to mRNA. But if you take a look at these life cycles, you'll be able to put it in the context of a real infection. It will make more sense to you. So this is an example of what's in the back in that appendix. It's a diagram of, remember, the infectious cycle, all the steps that occur. And you see here, this one involves some nuclear steps, which we didn't show last time for the RNA virus. So I suggest strongly that you have a look at these figures as much as you can. So let's take a general overview of viral genomes before we start looking at them individually. So for viruses with DNA genome, of course, the host genetic system is based on DNA. All the cells that these viruses infect have DNA genomes. And so these viruses, for the most part, emulate the host, especially in replicating their genome, but in also decoding it. But there are differences, because all viral genomes, or almost all of them, there are a couple of exceptions, are not like cell chromosomes. Cell chromosomes have a very specific structure. They're wrapped up with protein. They're made into nucleosomes. And most viral genomes are not made that way. They don't have centromeres. They don't have telomeres. So there are some tricks or differences that have evolved in viral DNA genomes with respect to the cell. And that's where the interest is, to find out how a virus is different from a cell, because that could give you a therapeutic way of intervening with infection. If you can find out what a virus does that's different from a cell, you might be able to find an antiviral drug that can inhibit that step without hurting the host. So this is of practical interest. So here are some questions that you might want to think about. I think these are important issues. We'll be learning about this information in the next half dozen lectures or so. I think these are important questions. First, how many ways can you think of to replicate DNA? Is there just one way, or are there a variety of ways? Do DNA viruses always replicate in the nucleus? You know, I said DNA viruses emulate the host. The host replicates its DNA in the nucleus, of course. Do all viruses do that? Most of the cells in the body are terminally differentiated, and they don't make DNA. What happens to DNA viruses when they enter these cells? Do they turn on DNA synthesis? Do they not care? Is it a problem? Do DNA viral genomes always encode their own DNA polymerase? Do they use that of the host always, or do sometimes they encode their own? And are there viruses with DNA genomes that encode no DNA polymerase? So the answer is yes, and you'll find out what they are. Finally, do DNA genomes exist that do not require a DNA polymerase to copy them? So you'll find the answers to all of these as we talk in the next 6 lectures or so. I think these are key questions about DNA genomes. Now let's take a look at each of these genome classes to acquaint you with what they look like and how they work. Starting with double-stranded DNA, there are 22 families of viruses. Remember, the families are the classification method where the name ends in viridae. So there are 22 families of viruses with double-stranded DNA genomes. And there are many viruses that infect different hosts. And the mammalian viruses are here: the adenoviridae, hepadnaviridae, herpesviridae, papillomaviridae, polyomaviridae, and poxviridae. Those are double-stranded DNA viruses that infect mammalian hosts. And right now they don't sound like anything to you, and they may make no sense to you whatsoever, but by the end of the course you'll know them, and you'll look at hepadnaviridae, and the replication cycle will pop into your head. The disease, how it causes disease, will automatically come out. The topology of the genome is critical. They're not all just double-stranded DNA. They can be linear, or they can be circles, and that determines how they are going to replicate. Topology has major implications for replication, as you will see. How do you make mRNA from a double-stranded DNA genome? What we call information extraction. Well, mRNA is produced when host or viral, can be either, depending on the virus. DNA-dependent RNA polymerase copies the minus strand of the double-stranded DNA. So this is an enzyme that makes mRNAs. In the cell, it's called RNA polymerase II. And that enzyme copies the minus strand to make plus strand mRNAs. Here's an essential fact. You cannot make mRNA from single-stranded DNA. It doesn't matter if it's minus-stranded. The enzyme will not recognize a single-stranded DNA template. So that's a key fact to remember. If you can remember that, it will help you a lot in that scheme, the Baltimore scheme. You can only make mRNA from double-stranded DNA. So that means for all single-stranded DNA viruses, the first thing that has to happen when the genome gets in the cell has to be made double-stranded. Otherwise, you can't make mRNA from it. So here's the information flow in a double-stranded DNA genome. Here's the double-stranded DNA here. So we have a convention in the book where the two strands are different colors of blue, different shades of blue. And I believe the dark blue is the plus and the light blue is the minus. And mRNA is a light green, a Kelly green, I think the artist called it, as opposed to olive green, which you'll see in a moment. So the double-stranded DNA can be directly transcribed to make mRNA. It can also be replicated, of course, and if that is carried out by the host DNA polymerase, you don't need any viral proteins to be made, but it varies depending on the virus. The mRNA is made into proteins, of course, and eventually you have new virions assembled by putting those proteins together with new genomes. So that's the information flow. It's quite simple, and it makes perfect sense if you know a few key facts. So there are two kinds of genomes with double-stranded DNA. We have, on the left, are circular double-stranded DNA genomes. These are the polyomaviruses and the papillomaviruses. Polyomaviruses are rather ubiquitous viruses which probably infect most of us. 80 to 90% of us are seropositive for one kind or another polyomavirus. And a papillomavirus is also ubiquitous in humans. The human papillomavirus is the causes of cervical cancer. They have circular genomes. They're rather small viruses. You can see the genome size is 5 to 8 kilobase pairs, pretty small. And so they don't have a lot of room for extra proteins, so they don't encode a DNA polymerase. They use the host's DNA polymerase. So these DNAs, so right away you know where's the DNA polymerase of the host. It's in the nucleus. That's where it works. So these viral DNAs have to get in the nucleus. Without me telling you the life cycle, you could figure that out. On the right we have larger viruses with linear genomes, linear double-stranded DNA genomes, and these are all bigger and they encode their own DNA polymerase. We have the adenoviruses, double-stranded DNA, the herpes viruses, also double-stranded DNA with complex repetitions of sequences, which Professor Silverstein will tell you all about, and the pox viruses, the biggest of the bunch here. This is double-stranded DNA, but the ends are joined together covalently, and that's why we call this a terminal loop. So in these other genomes, you have a free 5' and 3' end at each end of the genome, but for the pox, they're joined. And the significance of that we'll find out when we look at DNA replication. All right, the next class is double-stranded DNA, but they have a gap in the genome. So we call them gapped double-stranded DNA genomes. Well, actually, just gapped DNA, because it's not fully double-stranded DNA. So here it is. You see it's partially double-stranded. You have both colors of both shades of blue here, but there is also part of the genome that's single-stranded. So that's what we mean by gapped. So this is the strange genome of hepatitis B virus, which is also called the hepadnaviruses. It's a member of the hepadnaviridae. In addition to the genome being gapped, so it's circular, it's gapped, and we have a protein attached to the 5' end of the one strand here, the minus strand, the lighter blue strand. You see that's the one that's continuous up until the protein. Then there's a protein there. So there's a free 3' end and the protein linked to the 5' end. In addition, we also have a short RNA, which is covalently attached to the 5' end of the other strand. So one strand is complete, the minus strand with a little interruption, of course, and the other is about half completed, hence we have a big gap. So that's pretty strange. And in addition, as it exists, that cannot make mRNA. You should probably have been able to tell me that from what I told you earlier. Single-stranded DNA cannot be copied to mRNA. Even though there's a little bit of double-stranded DNA here, it won't work. So the first thing that has to happen is to fill in that gap, of course. Yeah?
Is it always the protein on the minus and the RNA on the plus?
Yes. It's always that way. And that's a byproduct of the replication, which we'll see. Yeah. Yeah.
How large are the relative double-stranded and single-stranded segments?
Ah, that's a good question. Half completed. So the single-stranded bit is about half here, and let's see. I do think we have a picture of the genome. No, I don't have the size here. What is it? The whole genome? I think that sounds small. I will. Anybody have a textbook with them? No. May look it up, and I don't know what the number is. So I will never ask you the size of the hepatitis B genome. It's not terribly big. It's not as big as these guys here. Okay. So you have to make this genome completely double-stranded in order to make mRNA. And to do that, you have to take off the protein in the RNA. You have to fill in the gap and make perfectly duplex DNA, and you have to make it covalently closed circular DNA as well. So all of this is a repair process that precedes mRNA synthesis. Now, in addition, this is a weird genome, and the reason it's weird is because it's made by an unusual enzyme, reverse transcriptase. It's a virus-encoded enzyme, which is very similar to the reverse transcriptase encoded by retroviruses. And retroviruses are viruses with a plus-strand RNA genome. They convert it to a DNA copy in their life cycle. But hepatitis B virus is a DNA gene. It's a DNA virus. Why does it need reverse transcriptase? Well, it works. I don't know why it does this. As you will see, the double-stranded DNA, the gap DNA is made fully double-stranded. RNAs are made from it. So one of those RNAs is an mRNA. It's not the same as this RNA here, which is a full-length copy of the genome. So this mRNA goes on to make protein. But this full-length copy of the DNA is then copied by this reverse transcriptase to make a DNA copy. And that's made double-stranded, and that's incorporated into the particle. So it's very strange because you already have a DNA. Why don't you just replicate it with a DNA polymerase, right? Doesn't do it. It makes this RNA and then reverse transcribes it. So no one knows why this is happening. It works. It's evolutionarily favorable because these viruses are still around. That's all we can say. So this step is carried out by host RNA polymerase. The copying of the DNA to make mRNA is done by a host enzyme. And then reverse transcriptase copies that RNA into DNA. It does not copy this mRNA into DNA. This one is not a full-length copy of the viral DNA. The protein is a byproduct of the replication of this virus, and it's a bit of a complex scheme, so we're going to go through that in detail, but it serves as a primer during replication. Okay, single-stranded DNA genomes. We have 5 viral families and 1 genus of viruses with single-stranded DNA genomes, and there are 2 with mammalian viruses, Circoviridae and Parvoviridae. So the basic problem with the single-stranded genome is what? Exactly. So you already know that. You've learned it. RNA can only be made by a double-stranded template, no matter what sense the single-stranded DNA. So we have to make these double-stranded before mRNA is made. DNA synthesis must precede mRNA production. So this is one of the questions I had up earlier. What's the order of events that happens? Well, here you have to first do DNA synthesis and then make mRNA. And that'll be different for different viruses. And by looking at the genome, you can deduce a lot of that. These genomes don't encode a DNA polymerase. They're small, as are the single-stranded, double-stranded viruses that we talk about. The double-stranded DNA viruses that we talked about. They use cellular DNA polymerase to replicate their genomes. And here they are, single-stranded DNA, made double-stranded by the host DNA polymerase. From that, we can make messenger RNA, and that is done by the host RNA polymerase. Messenger RNA goes on to protein, and the genome is then replicated by host polymerase to make single-stranded DNAs. Now you see something funny going on here. You're making both plus and minus DNAs, and mostly plus DNA goes into the virions, but sometimes you get some minus strand DNA going in as well. Here are the 2 kinds of viruses that infect mammals with single-stranded DNA genomes. With the Circoviridae have very small, among the smallest non-defective viral genomes. There are smaller ones, but they're defective. We'll talk about that much later. It's a circle, single-stranded circle, 1.7 to 2.2 kilobases. So there are not many proteins encoded by this, just a few. One is for the capsid, of course, just one, and a couple of others for replication. But these viruses are very strange. We don't actually, they cause infection of animals. So there's an agriculturally important disease of chickens. It's called beak and feather disease, which is caused by these viruses. So farmers are very familiar with this. Apparently humans are more than 90% seropositive for these viruses. We don't know what they do. We don't know if they cause any disease or if they're one of the viruses that infects everyone without causing symptoms. The Parvoviridae have a strange genome, linear, but the ends are hybridized in a way to make these T-shaped structures. And this will be relevant for DNA replication. These participate in the DNA replication process. An example of a human parvovirus is B19, which causes a childhood disease called fifth disease, which probably all of you have had. Yes?
So is the single-strand DNA always plus, or can it be minus as well?
I'm sorry, these...
If your diagram is always the first initial DNA single-strand.
So the minus will also work. It will be copied by the host, yeah. Also, if you have dogs, you know that they can be infected by parvoviruses and can kill them, so you should get them vaccinated. Yes?
How do these viruses prime DNA replication?
How do they prime it? We're going to leave that for Professor Silverstein, right? Who will tell you all the details of priming. Yeah, they have to be primed, absolutely, by primers. And remember, they mimic cellular DNA synthesis. RNA genomes, which are my personal favorite, only because I've worked on them for my career. And the RNA genomes are interesting because cells have no idea what to do with RNA. They have no RNA-dependent RNA polymerase, which could be used to copy the genome of an RNA virus. So that's another thing that you should remember. A cell can handle RNA. It cannot copy long RNAs. It can make small RNAs. It can make small interfering RNAs, microRNAs, that sort of thing. But it can't make a copy of a couple KB viral genome. So these viruses have to encode their own RNA-dependent RNA polymerases. These are novel enzymes which don't exist in the cell, and they're great targets for antiviral intervention because they don't exist in the cell. These polymerases copy the genome and they make mRNA. They do both. It can be the same polymerase or it can be different. We'll talk about examples of both. And of course, the mRNA that these polymerases make are readable by ribosomes by definition. So let's start out with viruses that have a double-stranded RNA genome. There are 7 viral families with double-stranded RNA genomes, and some of them are segmented. Some of them occur in one long double-stranded RNA piece. Some of them are in separate pieces. And a very well-known family is the Reoviridae. And these are double-stranded RNA viruses. They have 10 to 12 segments of double-stranded RNA. Included in this family are the rotaviruses, which are major agents of human gastroenteritis, typically happening in the winter. So now is the season for rotavirus gastroenteritis, often in settings like colleges and universities, daycare centers, elementary schools. You have an outbreak where half of the kids get gastroenteritis. It's rotavirus. There's actually now a vaccine against rotavirus because in a very young child they can dehydrate very rapidly as a consequence of the gastroenteritis. It can be fatal. There's another family, Birnaviridae, which infects invertebrates, and these viruses have 2 segments. Now, double-stranded RNA can't be translated by ribosomes. The only thing that can be translated by ribosomes is mRNA. So that's easy to remember. So double-stranded RNA doesn't work. So this has to be made into mRNA in some way. And that's what these viruses do. So there's the scheme of genetic flow, the flow of genetic information. We start with double-stranded RNA. And of course, the double-stranded RNA has a plus and a minus strand. So the minus strand is copied by a viral RNA polymerase to make an mRNA, which can be translated. So the ribosomes can't translate the mRNA that's present in this duplex. It's not accessible. So what these viruses all do is they copy the minus strand and make a single strand. And you'll see that it's a very interesting scheme that this is accomplished by. So now we have mRNA, and from there we can make proteins to make new virions, and that can be copied by the viral RNA polymerase again. All you have to do is now make a minus strand, make it double-stranded, and you have a new genome to put in the cell. And the reoviruses, of course, have the virus members of the Reoviridae have 11 double-stranded RNA segments. So for each of these, you have the same issue. You have to make first a messenger RNA for each one. And they're given names L, M, and S because when they were first discovered years ago, some of them were large, some of them were medium-sized, and 3 were small. Nothing fancy about that. Single-stranded RNA viruses, we'll start with those with plus polarity. There are 22 viral families with plus single-stranded RNA genomes. 8 of them have viruses that infect mammals and are significant pathogens. So a lot of the biggies are here. Picornaviridae, poliovirus, rhinovirus, Caliciviridae, another family of viruses that cause gastroenteritis. Often you may read online or in a paper that there's a cruise ship that has to come home because everybody's got gastroenteritis, and that's the virus that often does that. It's often shed by food handlers, and if they don't wash their hands, they will contaminate the food, infect everybody on the ship, and then they have to sterilize the whole ship before it can go out again. So those are caliciviruses that do that. Also, astroviruses are another important causes of gastroenteritis. We have the SARS coronavirus in this group. Flaviviruses, including yellow fever virus, West Nile virus, and hepatitis C virus. And we also throw in here retroviruses because they have plus-stranded RNA genomes, but they're from another Baltimore group, of course, because they have a DNA intermediate. And finally, the togaviruses, which include rubella virus, a childhood infection, and viruses of various sorts that cause infections of the brain, encephalitis. So we'll talk about a number of these in detail as far as replication goes and pathogenesis because they cause interesting diseases. So the single-stranded, plus-stranded RNA viruses have genomes of a variety of sizes. They range from the biggest, 28 to 33 kilobases of the coronaviruses. Those are the biggest known RNA genomes. They don't get any bigger than that. And some of the others are smaller. And we don't know why the size of these RNAs is limited. You know, DNA genomes can be over 1 million base pairs in length, but the RNA is only this long, and we don't really understand why. Many of these viral RNAs are messenger RNAs. They can be translated in the cell. And so you see this 5' cap structure is a feature of an mRNA needed for translation, and we'll talk about that separately. And some of these have 3' poly A stretches in their genomes as well. So these plus-stranded RNA genomes are translated directly into protein by host ribosomes. So these are all not only viral genomes, but they're messenger RNAs. As soon as these are released into the cell, the ribosomes attach to them and start making protein. So nothing else has to happen. We've talked about a few other genome configurations where you have to either make double-stranded DNA or make mRNA from double-stranded DNA or fill in a gap. But for these viruses, as soon as they get in, they can be translated. Right? Nothing else has to happen. Now, so there's one family of viruses, of course, of this plus-stranded RNA, single-stranded, where there is a DNA intermediate in the life cycle, and those, of course, are the retroviruses. There are 2 significant human pathogens in this family so far, maybe more: human immunodeficiency virus and human T lymphotropic virus, HTLV1. And the strategy is very unusual. The viruses have a plus-stranded, single-stranded RNA. It's converted to a minus-strand DNA by the viral enzyme reverse transcriptase. And it's called that because when it was discovered in the early 70s, up to that time, the information flow was thought to be only DNA to RNA in all cells and all viruses. So when these were discovered, they said, it's going in the reverse direction, so they called it reverse transcription. So I always like to wonder what would have happened if these had been discovered first. So then we would be reverse transcribers, right? But that didn't happen. So they make single-stranded DNA negative polarity from the plus RNA, and then that's made double-stranded by the same reverse transcriptase, so it can also copy DNA to DNA. And then from the DNA, the virus can make mRNA for proteins, and it can make RNA, which is a new genome. Right? That's that genome that started out. It can make new copies of this. And these two are not the same. So this is not an mRNA, even though it's a plus-stranded RNA. So I just said it's not an mRNA, but now I'm contradicting myself. It's an mRNA, but it's never translated. It's a plus strand, but it's never used as a message. So in theory, when this mRNA gets into cells, it could be translated because it's plus polarity and it has all the initiation and termination sequences that are needed, but it's not. It's not translated. And we're not sure why, but that's the observation. It's converted to a DNA copy, and then the DNA is transcribed by a host DNA-dependent RNA polymerase to make the messenger RNA. This double-stranded DNA integrates into the host chromosomal DNA and becomes a permanent part of the host's genome. We call that a provirus. So the viral DNA integrates into your chromosome. It's now called a provirus. It's permanently there. You can't get rid of it unless you die. And it's from within the genome that the mRNA is made. So the host DNA-dependent RNA polymerase transcribes this double-stranded DNA as it's sitting in chromosomal DNA, not as it's floating around in the nucleus, but only if it's integrated into the host genome. And all these processes we're going to talk about in great detail. So the proviral DNA serves as template for viral mRNA and genome RNA synthesis. The RNA polymerase, as I've told you, copies the proviral DNA to make mRNA. Some is translated into proteins, and some is packaged into virions. So let's go to the minus strand RNAs now, single-stranded RNA minus sense. There are 7 virus families with these kinds of genomes, and again, more pretty serious viruses here. Paramyxoviruses, measles, mumps viruses are here. Rabies virus. Bornaviridae, a virus of agricultural importance, infects horses, and turns out that people seem to be infected with this virus as well. And just a few weeks ago, a DNA copy of this RNA virus was found to be integrated into the human genome. And we don't know why that's there, because it's not a retrovirus, but we'll talk about that later. Ebola and Marburg viruses are here. These are highly lethal, but pretty rare infections. Influenza virus and Lassa virus. So they start with a minus strand RNA, which you know can't be translated, so it has to be copied to a plus strand messenger RNA. And you know that the cell can't do that, so this has to be done again by a viral enzyme. And the mRNA can make protein, which can make new virions. In some cases, the plus RNA and the mRNA is the same, but often it is not. These are 2 separate molecules. Okay? So you have 2 separate processes, one making mRNA and one making RNA that's going to go on to make new viral genomes. So the genomes can't be translated because they're minus strand. You have to copy them. And the cell can't copy it, so the virus has to do it. Well, here's the conundrum. The RNA can't be translated, right? The minus strand RNA can't be translated to make that RNA polymerase. So what has to happen? The virus has to bring the enzyme in the cell with it in the particle. So these negative stranded RNA viruses, besides having a genome in the virion, they have enzymes that are used as soon as the RNA gets into cells to make a plus strand copy. Okay, so when you need to copy the minus strand, which you have to do by definition, has to be done by an enzyme brought into the cell in the virion. There are no cells in the enzymes in the cell that can make mRNAs, so the viral enzyme does that, and it also replicates the genome. And I've told you all of this information. So here are what the genomes look like. These minus strand RNA genomes can be either in pieces, which we call segmented, which is shown up here, or they can be non-segmented. So the most famous segmented RNA viruses, negative strand RNA viruses, are the influenza viruses, which are in the family Orthomyxoviridae. They have between 6 and 8 RNA segments. The paramyxoviridae, measles virus, mumps virus, rabies viruses have single RNA molecules, just one RNA that is negative stranded. So 2 different configurations. Now, if you have a genome that is in pieces, like influenza, that has big consequences for genetic variability. So, for example, if you have 2 different influenza viruses, when they infect the cell is filled with all the 8 RNAs of both viruses. So the new viruses that are produced can pick up an assortment of RNAs from both parental viruses. So this is called reassortment. And this is one of the reasons why we have a pandemic strain arising every 20 or 30 or 40 years. And we'll go into this in more detail later on. Okay, now some viruses that are actually classified as negative stranded RNAs are in fact ambisense. They have both plus and minus RNA, and the arenaviruses and the bunyaviruses are like this. So what do I mean by ambisense? Well, here are the genome segments of a bunyavirus. This virus happens to have 2 different genome RNAs, an L and an S. And here's the olive green, that's minus stranded, and the Kelly green is plus stranded. So you see one of the RNAs, LRNA, has both plus and minus strand polarity. So all that means is that the plus part can be translated right away and the minus strand can't. And there's the second RNA, the sRNA, is the same. So what happens when these viruses infect the cell, so the RNA gets into the cell, the very 5' part can be translated because it's plus polarity. So this is translated into protein. But this part here cannot be translated. It's minus strand. So a separate messenger RNA has to be made complementary to this negative stranded region. That's shown here. It's a plus stranded mRNA. and that can be translated into protein. Okay, so that's why we call them ambisense. And again, why this arose is anybody's guess. It just my view is it works, so it's remained around. So there's a great website that has really nice illustrations of all this that I've told you. And here's the URL here. It's a Swiss website. And so here's one of the pages where they have the Baltimore classes that I've just told you about. And you can click on any of these lozenges up here, and you go to pages where they have lots of additional information about all these viruses. They have great artwork on this site as well. Okay, what do viral genomes look like in the virus? Do they look like this? No, this is something we draw in textbooks, in which I show you here, but that's not what the genome looks like. It's only for illustrative purposes. And I just want you to understand that the genomes are much more complicated. They look something like this. So this is a picornavirus plus stranded RNA genome, which has been analyzed with a computer algorithm to look for parts that could be folded, which are energetically likely to be folded. And we'll see later on that RNA often folds into extended stem loop structures. And that's what this genome has done. Here's the very 5' end of the genome here. And it's the whole entire sequence of the genome, 7,500 nucleotides, is folded extensively into stem loops and secondary and tertiary structures. And there's some evidence that this actually happens. So probably it's biologically relevant. We'll talk about some examples of stem loops that are relevant for specific processes. But I want you to remember that genomes are not just lines, squiggly lines of olive and Kelly green, but they have a really extensive folding. And there are all kinds of features of genomes, both RNA and DNA, that are unusual. Some of them are summarized here. We've already seen the termini of the parvovirus DNA, the funny gap in the hepatitis B virus, while some RNA genomes have interesting structures at their ends and in the middle that are biologically relevant. As you'll see, retrovirus DNAs are actually retrovirus RNAs are actually dimers in the virion, hybridized to a tRNA. And there are lots of very interesting stem loop structures in various genomes which have functional properties. So they're not just straight lines. Now, what can you do with genomes? Well, you can do genetics. And so we need to have a few definitions out front initially. Wild type we're going to use a lot in this course. Wild type simply means a virus that you start with. It's usually a virus you have in your laboratory, and you start with that and you make mutants of it. It doesn't mean that it's a virus that you got from nature. In fact, it could be very different. Viruses that we get from animals or people are called clinical isolates or field isolates, and they may be different from the wild type that you're working with in the laboratory. Another definition that we need to get out front is DNA-mediated transformation. That is the introduction of foreign DNA into cells. So it has this long word because of historical reasons, which I'll tell you, but you have to be very careful not to confuse it with oncogenic transformation of cells. So you always have to have DNA mediated. You can't just say, I transform the cells, because no one will be sure what you're talking about. Okay, so always DNA mediated transformation. The reason it's called transformation, it comes from this 1944 paper, which showed that the DNA of a pneumococcus of one strain can be inserted into a different strain and confer a phenotypic difference. So this was done by a group of scientists here at the Rockefeller in New York. It's the famous Avery McLeod-McCarty experiment. So they have pneumococci with two different colony morphologies, and they showed you could take the DNA from one and put it into the other, and it would transform the one colony into the other. So that's where the transformation came from, and it was DNA mediated. So here's what they did. They had pneumococci on an agar plate, two different colony morphologies, and they could turn this one into this one by putting DNA of this one into this one. So they called it transformation of pneumococcal types, and that became DNA-mediated transformation. We have another word that is now being used to describe this called transfection. Transfection is the production of infectious virus after DNA-mediated transformation of cells by viral DNA. This was first done with bacteriophage lambda. So you can imagine you take lambda DNA and you put it into cells by a process called DNA-mediated transformation, and the virus reproduces in the cell. So that was called transfection because it's transformation combined with infection. Transfection. But nowadays, everyone uses transfection because they don't want to say DNA-mediated transformation. It is a bit of a long word, so they shorten it to transfection. But unless you're talking about viruses, it's not right. But you know how the English language is. It gets molded to what everyone wants. So to avoid this is futile. So I want to tell you what the correct terminology is. Transfection only has to do with viruses, but people use it for other things. When we talk about genetic methods, we talk about mutations, which is a change in DNA or RNA. You have base changes, deletions, insertions, rearrangements of different sorts, right? Nonsense, missense mutations. But you don't make a mutant protein. You don't make a mutation in a protein. By definition, a mutation is an alteration of a nucleic acid sequence. So what method allowed the application of genetic methods to animal viruses? So this, now we're talking about, 1950-ish. What method do you think allowed us to start studying the genetics of viruses? Anybody have a clue? We talked about it last time. No? What's that? So you're saying it. Plaque assay. A plaque assay, remember, lets you purify your populations of viruses. So you can pick a plaque, and if you've mutagenized this virus, you can have a virus with a specific mutation. Before this, you couldn't do genetics of viruses. So 1951, Renato Del Becco not only allowed us to quantify viruses, but also to do genetics. Now, back in the 50s and 60s and even 70s, the way people made mutants were very different from the way we do it now. Let me just tell you that briefly because it's good to know this. Now, as we will find out later on, RNA viruses are the masters of mutation. Their polymerases make a lot of mistakes, one misincorporation in every 10,000 to 100,000 nucleotides that they polymerize, and they don't correct their mistakes. Okay, so they are the masters. DNA viruses have a higher misincorporation rate because their DNA polymerases have error correction machinery. RNA viruses don't. So you could just take a stock of an RNA virus and look for a phenotype in it. If you wanted to, you could probably find it. But a lot of scientists used chemical treatments. They would treat the viruses with various chemicals to make more mutations than would naturally occur. And these were mutagens like nitrous acid, hydroxylamine, alkylating agents. They modify the viral DNA or RNA and cause changes in base pairing. You could also use base analogs, UV. A variety of treatments were done to introduce mutations. These would be introduced at random in the genome, and then you'd have to figure out where they were and what kind of changes they made to the virus. So you would look for phenotypes. A phenotype is the expression of a genetic change. You could look for temperature sensitivity, the inability to grow at a high temperature. Size of plaques. A mutation could make small plaque size. That was often done to track mutations. Cold sensitivity. The virus doesn't grow at low temperatures. Resistance to drugs, escape from antibodies, infecting different host cells. So these were just some of the phenotypes that virologists looked for in the old days when they would randomly mutagenize a genome and then say, where is the mutation and what does it do? But nowadays, our life is very much easier because we can engineer specific mutations into genomes. And that's because we have what are called infectious DNA clones, which we can change. We can modify the DNA any way we want. We can make single base changes anywhere, and then we take the modified DNA, we put it back into cells by transfection, and we get mutant viruses out. And this is a modern validation of the Hershey-Chase experiment of 1952. Okay, the Hershey-Chase experiment, well, I'll show you in a moment, proved that the DNA of a phage was the genetic material. And this does the same thing because you're actually purifying the DNA and putting it back into cells and getting viruses out. So you can make all kinds of mutations by modifying DNA. And just to remind you, the Hershey-Chase experiment showed that when you label the viral DNA of a phage, the viral DNA gets inside the cell. And then the new phages that are made have the labeled viral DNA in them. So they said this means that the DNA is genetic material. And a lot of people doubted that for many years, but now we know for sure because we can take that DNA and put it into cells and we get infection. So we have infectious DNA clones for just about all viruses, both DNA viruses and RNA viruses. For a lot of these DNA viruses, they're already double-stranded, so it's very easy to put them in a plasmid vector and grow them in bacteria. And for RNA viruses, you can convert them to DNA using reverse transcriptase. You can clone the DNA in a plasmid and then reintroduce that into cells. Now let me show you two ways to make a virus from a DNA copy of an RNA genome. So the first one is poliovirus. If you take the RNA out of poliovirus, if you purify the RNA and transfect it into cells, you will initiate an infection because the RNA has all the information to guide an infectious cycle, and it's mRNA, so it's translated as soon as it gets in cells. If you make a DNA copy of this mRNA and put it in a plasmid, you can either take the plasmid and put it into cells or make mRNA from this plasmid in vitro using an enzyme and put that into cells. In either case, you get virus made. So now you've made a DNA copy of poliovirus, and you can make changes in it and then recover viruses with those changes. So this works very nicely for plus-stranded mRNA viruses. For other viruses with negative-stranded genomes, the approach is a little more convoluted. It took many more years to develop, but this is an example of how you do it for influenza. So influenza virus genome is 8 negative-stranded RNAs. And if you put a negative strand RNA in a cell, nothing will happen, right? Because the cell can't do anything with it. So what you have to do with influenza is you have to make a plasmid for each, which has a copy of each of the 8 negative strand RNA segments. So this is a double-stranded DNA plasmid. And it has a promoter at each end so that the one plasmid for each RNA will give you both mRNA to make protein and negative-stranded RNA to make more genomes. So you make 8 of these plasmids, one representing each of the 8 negative-strand RNAs. And you take all 8 and you put it in a cell and you get influenza virus out. Okay? And so similar approaches are used for other viruses as well. And this was what was done to resurrect the 1918 influenza virus strain. So back in 1918, we didn't know what influenza virus was. It hadn't been isolated. It wasn't isolated until 1933. So we don't have the virus to study. But a number of years ago, 2005, RNA was isolated from paraffin-embedded lung tissues and from an autopsy of a victim from 1918. So the Army had stored lots of lung samples from soldiers who had died from influenza. These are on slides embedded in paraffin. So what they did is they took the tissue off the slide, extracted the RNA, and sequenced it. And they put together the sequence of all 8 segments. They also found someone who had been buried in the permafrost, forgot where it was, since Alaska. So the ground is frozen all the time, and they went to a grave where this person had been buried since 1918, and they dug the grave up. They did a biopsy of the lung, and they went back and they sequenced the RNA. So basically, they reconstructed the whole sequence. They made 8 plasmids. They put it in cells, and they got the virus back. So we now have the 1918 influenza virus, which we didn't have before, thanks to this technology. All right, what else can you do with... So you can do great experiments in the lab with infectious DNA clones, but you can also make virus vectors to treat diseases, which is very exciting. So you can do gene therapy. You can modify a virus to carry a gene that you want to give to people who are missing the gene. So in many cases, you take cells out of the patient, you infect them with a virus that has the gene that's missing, and then you put the cells back into the patient. You can deliver also antigens by this approach. You can make vaccines using viral vectors. So a lot of the HIV vaccines that are being tested are actually HIV proteins being expressed from an adenovirus vector. And you can do all this because you can manipulate the DNA of these viruses and recover virus after transfection. We also, of course, have research uses for these vectors. So viral vectors have very specific requirements, which is why progress has not been terribly fast. First of all, yes?
I was wondering if specific cells are best for this purpose?
It depends on the disease. So for some diseases, you take bone marrow cells and you transduce them with a vector and then put them back in, depending on what cell type you're targeting. For cystic fibrosis, for example, the cell targets the epithelium of the lung, so you can't do that. So you have to put the virus directly into the lung. So one consideration is how much foreign nucleic acid you can put into these cells, virus genomes. That limits it. Also, the viruses can't kill you anymore, right? There's no point in giving you gene therapy with viruses that are lethal, so you have to eliminate it, and that's not very easy. And you have to have the promoter driving the gene that works in the tissues that you're interested in. So, bless you. So because of all these problems, it's been very slow, but there have been some progress made. These are just some of the genetic diseases that might be treated using viral vectors containing human genes. And some of the well-known ones include cystic fibrosis, which is a defective salt transporter in the lung. So here's the disease and the defect and the incidence. And these are the various target cells that you would want to make sure that the vector goes into. So there have been a couple of trials with a variety of vectors, and there have been some problems. So for example, using an adenovirus vector to treat cystic fibrosis. A young man, I think an 18-year-old in Pennsylvania, died very shortly after the infection because he had a massive immune response against the vector. In France, some gene therapy trials were being done using retroviral vectors, which integrate into your DNA. And in a couple of the recipients, it integrated next to an oncogene and caused tumors. So a lot of unexpected consequences, which you have to work your way through. But there have been some successes. This is just an example of how to make an adenovirus vector. You take the double-stranded DNA genome, you put the gene of interest here, and then you cut out most of the rest of the genome. So this is basically a defective virus, which you have to give capsid proteins in order to be packaged. And once you're infected with this, it will express the protein, but it will not replicate and make more viruses. So that's a very important consideration. Retrovirus vectors are very popular for gene therapy. The retroviral genome looks like this. This is a double-stranded DNA copy of the retrovirus genome showing where all the proteins are encoded. You can actually cut out most of this coding region and just leave these two end sequences called LTRs, and we'll talk a lot about this later. You can put a gene of interest right in here, and as long as you provide the other proteins to the virus, it will replicate. So for example, and you can separate these genes on different plasmids. So for this, for example, the core genes make the capsid. The enzymes include the reverse transcriptase, which the virus needs to replicate. And the envelope is the glycoprotein on the virion surface that's needed for attachment to receptors. So if you take two plasmids, one encoding the capsid proteins and one encoding the envelope proteins, and put those in cells, you will get empty retroviruses. So the full capsid is made without a genome. So all you have to do is put in your gene of interest into a third plasmid. You now get a retrovirus with your foreign gene. These will infect cells, but they will not replicate because they're missing a lot of the key proteins. So retroviruses of these sorts are used often for gene therapy, and especially retroviruses related to HIV because they can infect cell types that other retroviral vectors can't.
Once you get the empty capsid, can you just insert the DNA you want into it, or do you have to do it with the second plasmid?
That would be difficult to do that. Yeah, it would probably be very inefficient. You just do this, and this still is a defective virus, and it's not going to be a problem. So I just want to leave you with this one blog post, which is a very recent success in using a lentivirus gene therapy to treat a very rare neurological disease called X-linked adrenoleukodystrophy, which is a neurological disease caused by a defect in a transporter, a membrane protein that transports things in and out of the cell. So what they did, they found, this is usually treated by transplantation, but you have to have a donor that matches you. And they had a couple of children in this case who they didn't have a match for. So they took out their bone marrow and they infected it with a retrovirus made in the way I just told you, containing the normal transporter. And then they destroyed their own bone marrow by irradiating them and then injected them with these virus-infected bone marrow cells. And these repopulate them and give rise to all the cells that come out of the bone marrow. And this actually worked in these two cases. Their symptoms were much less than would have been expected. So this is a very difficult approach because it's basically a bone marrow transplant. And so it's costly and risky, but it can work. And this is just 2 kids, but it works, and it shows that the approach might be useful in the future.